So welcome back again. Let's take a look at our concept check questions and see what the answers to these are. If you made a chocolate lab, which is the homozygous recessive, with a black lab of unknown genotype, so this is a test cross, the resulting litter has only black puppies. Do you know the genotype for the black parent? Well, here is our cross. Oops. We've got our black parent, which is big B question mark. It is definitely an unknown. And we're crossing it with the little b, our chocolate lab. And we're told that all the parents, all the puppies are black. So if all the puppies are black, the only way that they could be black is if they get a big B from the black parent. But if the black parent had a big B and little b, in other words, it was heterozygous, then we would expect some of the puppies to be chocolate because they're all getting a little b from their parent, from this parent. So the only way that they could potentially get the big B from both parents is if this individual is big B, big B. So yes, you do know the genotype of the parent. What is the probability of getting an ace in a 52 card deck of cards? Well, an ace, how many aces are there in a deck of cards? Are you a poker player? Are you a card player? If you are, you know that there are 4 aces in the card, so it would be 4. Let's do it this way. So to get an ace, it would be 4 out of 52, and that reduces to 1 out of 13. So that would be your probability of getting a single ace. Now, if you have an ace and a king pair, well, getting an ace, we already know, is 1 out of 13. And how many kings are there in the deck? There are 4. So the king would also be 1 out of 13. And if we want the ace and the king pair, we need the ace and the king. So that's going to be our law of multiplication. So it's going to be 1 out of 13 multiplied by 1 out of 13. I'm not sure what 13 times 13 comes to, but whatever that comes to, it's going to be 1 out of, I'll do it this way, 1 out of 13 squared, because we're going to multiply them together. 2 aces. Well, that's going to be same as it's going to be the same as this. So it's going to be 1 out of 13 squared. Not good odds. That's why you need to be really careful when you play poker. And you have to remember what the odds are of getting a particular combination of cards. And it's not just what is the chances of you getting a particular combination of cards, it's what are the chances of other people in the game also trying to get particular combinations of cards. Not an easy thing to be able to predict. But again, winning at poker, it's all about being able to predict the possibilities and to be able to do simple statistical math in your head and be able to calculate the chances of you getting the combination that you need, of getting a winning hand versus someone else getting the winning hand. So The next section focuses particularly on human genetics and human inheritance patterns. And one of the problems that we have with looking at human genetics patterns is simply it's very difficult to do genetics experiments on humans. In fact, I would argue that it is just unethical. We don't decide who should marry who and who should have kids with who. That's not a society that I particularly would want to live in, and I'm pretty sure it's not a society that you would want to live in either. It has been tried in the past in Nazi Germany, in the Soviet Union, and in other repressive societies. This sort of thing has been tried. I don't know whether that's attractive to you. It's certainly not attractive to me. It's not something that I would want to participate in. The second issue with doing genetics experiments in humans is that our generation time, the time between one generation and the next, is very long. We usually say it's either 20 or 25 years between one generation and the next. That's a long time to wait for the results of your genetics experiments. And number three, we don't reproduce as much as we ought to make our data statistically valid. The more offspring that you have, the more data you have, the more valid are your genetics experiments and the results of your genetics experiments. And even when humans were having large families, we still weren't really producing enough offspring for there to be statistically valid results. So all of these observations, and particularly the first one, which is, I mean, after the first one, we just stop right there. Even if we have other objections, just number one ends the discussion right there. But that means that it is very difficult then to take a look at genetics inheritance in humans, except it's not. We do this all the time. And the way we do this is by doing what is referred to as pedigree analysis. We look at family pedigrees. We look at the way in which human traits are passed from one generation to the next. And as it turns out, you really don't need a lot of information to be able to figure this out. If you've got data for 3 generations, you've probably got enough to establish the inheritance pattern for a particular genetically determined characteristic. So here are some traits that we know are genetically determined in humans. And these are what are referred to as superficial traits and neutral traits, meaning they do not affect our fundamental humanness. They are what make us different in physical appearance. That's it. They don't change our fundamental humanness. They don't affect our ability to thrive as a human. They are neutral traits. They don't have any effect on your survival and your opportunities to reproduce. They are neutral traits. They're not an advantage. They're not a disadvantage. They just are. So you can see the distribution of these traits. Now, one of the things that you might notice is that the individual who has freckles also has red hair, as do a number of other individuals in this slide. But one of the things that we do note in humans is that if you have red hair, you very frequently also have freckles, and you also very frequently have green eye color. There is a reason for that, which we will get into a little bit later. But just store that in the back of your mind for now, and we'll come back and revisit that one in a little while, a little bit later in the presentation. So, given that these traits that we've just looked at are traits that we can track in family pedigrees, basically family trees is another way of describing this. We can do the same thing for essentially any human condition that we think has a genetic basis. And by tracking it through at least 3 generations, maybe more, we can determine what the inheritance patterns is. Is it a recessively inherited trait, or is it a dominantly inherited trait? Because both occur in any group of organisms, including humans. So here is a pedigree analysis, or a family tree. And you can see that individuals are identified as male or female on the basis of the shape. Square is male, round or circle is female. If the square is colored, that's an individual who has the recessive trait, and it's the recessive trait that's easiest to follow, because we know that the recessive, in an individual, remember, an individual with recessive phenotype has to be homozygous for it, so it's real easy to follow recessive traits. And you can see how this is passed from generation to generation in this family. Now, why would we do this? And again, free earlobes versus attached earlobes. This is another one of those superficial traits. It is our particular personal collection of these superficial traits that gives us our unique physical appearance, our surface appearance, and separates us from the other individuals around us, including the members of our family, because every member of our family will have a different combination of these superficial traits. It's what means that we don't all look like the same identical grey blobs. Make life kind of boring if we were all the same identical grey blob in terms of our appearance. Again, remember, these are surface traits. They don't affect our underlying humanness. They just make us different enough that we can separate individuals by what they look like. It doesn't affect anything else. So this is a pedigree analysis, in this case, for attached earlobes versus free earlobes. Free earlobes being the recessive trait, attached being the recessive trait, and free being the dominant trait. And you almost had it backwards. So you can see how this is passed down through the generations from one generation to the next. And we can then make a prediction on the basis of what we see in this family tree of who has what particular version of the trait on the basis of what we see the inheritance pattern to be. So when we start thinking in terms of inheritance patterns, we also need to remember that gender is a product of chromosomes. The fact that we have this pair of chromosomes, the X and the Y chromosomes, that determine gender. The XY combination being male, the XX combination being female. And as we'll see, this difference in size between the X and the Y chromosome, and the fact that the X chromosome has a large region that is simply missing in the Y chromosome, does have consequences. There is an inheritance pattern that is associated with specifically the X chromosome. We refer to it as X-linked, as you'll see. So here are some traits that are known, or some genetic conditions that are known to exist in humans, and the
way in which they are inherited. So albinism, cystic fibrosis, phenylketonuria, sickle cell disease, and Tay-Sachs disease, these are all inherited in a recessive fashion. In other words, you have to be homozygous recessive for these. Achondroplasia, Alzheimer's, Huntington's, and hypercholesterolemia, or what is referred to as familial hypercholesterolemia, inherited, in other words. These are all inherited in a dominant fashion. You only need a single copy of the allele to show the trait, because they are the dominant trait. So cystic fibrosis is found specifically amongst people of Northern European origin, although we believe the allele may have originated in the Indus Valley in India and Bangladesh, because it seems to give you some sort of protection against cholera. Sickle cell disease traces its origin to the malaria zone of Africa, but there are equivalent genetic conditions that are found in other malaria regions, particularly in southern Nepal and northern India. And these allele, or the sickle cell allele, in an individual who is heterozygous, actually provides them with protection against malaria without the burden of having full-blown sickle cell. Tay-Sachs traces its origin to a group of individuals known as Ashkenazi Jews. These are Jews that trace their heritage to Eastern Europe, so Poland, Russia, that area of Europe. So why should these conditions have a particular geographic origin? In part, it's because they do actually provide some advantage in the geographic region where they originated. I've explained with sickle cell and cystic fibrosis. Not sure about Tay-Sachs in terms of what advantage it might have had in its geographic region of origin. But also notice that I said it also is associated with particular ethnic groups. And the reason why is because individuals of an ethnic group tend to marry within that ethnic group. And if there is a genetic disorder, particularly one that's recessive, not so much one that is dominant, but one that is recessive, then the chances of two individuals within that ethnic group being heterozygous for these particular alleles is probably quite high. And if they marry and have children, then they have a 1 in 4 chance of producing a child with one of these conditions. This is the reason why these conditions have persisted in human populations and have typically been associated with particular ethnic groups. That is not necessarily the case anymore, because as people have become more mobile and people have the freedom to marry outside of their ethnic group of origin, they also carry their alleles with them, and those alleles have become more widespread. In the case of dominant disorders, these tend to get eliminated because individuals with many of these dominant disorders, particularly Huntington's disease and achondroplasic dwarfism, have a shortened lifespan. There are also some unfortunate side notes, particularly to Huntington's disease, as you'll see. But natural selection has a tendency to eliminate dominantly inherited disorders simply because they are a disadvantage. And if they reduce an individual's lifespan and reduce their opportunity to reproduce, then the chances are that their alleles will ultimately be eliminated from a population. That is simply the way evolution operates. Let's take a look at the way some of these characteristics are inherited, focusing first on the recessive and then looking at the dominant. So in order to show a recessive trait, you need to be homozygous for it. That means that your parents both have to be heterozygous for it in order for you to get a copy of the allele from each parent in order to be, so that you become homozygous for it. Consequently, we introduce the term carrier. So the term carrier is defined for you here. It's an individual who carries the recessive allele, so they have the allele for the recessive trait, but appear normal. In other words, they carry the allele for the trait, but they don't show the trait. And in order for so for a child to show one of these recessive traits, both parents must be carriers. And so this is the reason why individuals who are closely related are advised not to get married, even though historically those sorts of things have happened. After all, Charles Darwin married one of his cousins, and he felt really guilty about that for the whole of his life because some of his children had some issues, and he blamed himself for having married his cousin as a reason for these sorts of things happening. This is why some of these recessive disorders have been in the past associated with particular ethnic groups, because again, if there's a relatively high frequency of the recessive allele within the amongst the individuals in that ethnic group, and they tend to marry within the same ethnic group, then the chances of two individuals being carriers for that same recessive trait is pretty good. And therefore, there is the possibility of producing a child with the condition. But also, if you do the Punnett square, you'll see that these parents also have a very high chance of producing children who are themselves carriers for the allele. So it propagates the allele in the population by increasing the likelihood of individuals who are heterozygous. So this is the nature of the inheritance pattern for these conditions. So here is again another situation. So here are two individuals who are heterozygous for the hearing allele and the deafness allele. And so here is their child who is deaf, the homozygous recessive, but notice that they also have a 1 in 2 chance of producing a carrier child, which continues the presence of the allele in the population. So when we're thinking in hearing versus being deaf, well, we know people can compensate for deafness, and there are ways of compensating for that. But what about things like Tay-Sachs, which is lethal, sickle cell, which has all sorts of problems associated with it, as we will see, and shortens lifespan. Same with cystic fibrosis. If you have two parents that are normal but are carriers for the alleles, yes, they have a 1 in 4 chance of having a child with the condition, but they have a 2 in 4 chance of producing carrier children that maintain the presence of the allele in the population. This is the nature of recessive alleles, unfortunately. So we've talked about cystic fibrosis and the nature of cystic fibrosis, so I'm just going to move on from there. With dominant disorders, the presence of a single allele means that the individual is going to have the disorder, obviously. Now, in the case of achondroplasia, the situation is a little bit more complicated in that the big D combination this is our lethal combination. So any embryo that is produced with a big D combination, that embryo dies in the uterus. It's probably eliminated through a miscarriage or a spontaneous abortion, as it's called in the medical world. So you don't see the big D combination here in the Punnett square because it would be fatal. And those embryos simply are not carried to term. So you do have the big D, little d combination. These are the achondroplasic dwarfs. It is a dominant trait. So having the big D is the, it leads to the achondroplasic dwarfism. And then little d is the normal characteristic. So here is something that is interesting, and it may not strike you immediately, but dwarfism is the dominant trait, yet the vast majority of us show the recessive trait. Interesting, right? Because under normal circumstances in Mendelian genetics, we would expect the dominant trait to be the one that's present most frequently. But we know from this situation that in fact the recessive trait is the one that is present in the greatest number. And there's a simple reason for this. So what do I mean by this? So this is the drawback for achondroplasic dwarfs. Yes, it's the dominant trait, and you would think that that would be the one that is most frequent, but being an achondroplasic dwarf means that you also have a very shortened lifespan, typically less than 50 years. And that's because you have a small body, and it's typically the arms, the legs, and the trunk that are small. The head is normal size, but you're also trying to fit normal sized body organs into that shortened trunk. And that never works out well. And so people with achondroplasic dwarfism do have a shortened lifespan. And it's substantially shortened. And so having normal height is the favored trait. You tend to live longer, you tend to reproduce more frequently, you leave more offspring behind as a general rule. That's natural selection at work. And so this is the reason why many of these dominant traits, particularly if they show up early enough in the life of the individual, will ultimately be eliminated from the gene pool. The same is true with Huntington's disease, although Huntington's disease has a very cruel twist. And that is that the condition does not begin to manifest itself until the individual is around 30 to 35. And it is terminal and it is lethal. It is a neurodegenerative disease, and it does kill the individuals who develop the condition. And if you have a single copy of the allele, you will develop the condition and it will kill you. But typically, the emergence of the condition doesn't begin until middle age, around 30 to 35. And what have you done by that time? Already had your kids. And there's a 1 in 2 chance of you passing that allele on to your children. So there's a 50% chance of them getting that allele and therefore having Huntington's disease themselves. And so for many years, it was a matter of great thought on the part of individuals where Huntington's disease was known to exist in the family to decide whether you wanted to have kids or not. Because at that point, at the time that you were deciding to have kids, you didn't know whether you were going to develop Huntington's disease or not. And there was no way of checking for the presence of the allele. That's all changed now because we do have a genetic screening test that allows you to identify whether you have the allele or not. And so you can make a decision on whether you want to have children or not. But up until about 20 years ago, there was no way of knowing whether you had the allele and whether there was a 50% chance of you passing it on to your kids as a consequence. So this is again, the fact that these dominant traits are inherited as a consequence of having a single copy of the allele does not mean that the condition can't turn up. It's just going to be very, very infrequent. If you want another example, you know that we are, or the vast majority of us, have 5 digits on each hand and each foot. That happens to be a recessive trait. The dominant trait is to actually have 6. It's a condition called polydactyly. What is interesting is, again, the vast majority of us are homozygous recessive, only having 5 digits. Having that 6th digit, that's a dominant trait. And so it's inherited in a dominant fashion. You may not be aware of individuals who have this. Typically, early on, surgeons remove the extra digit. It's usually located on the side of the hand over here by the pinky or next to the little toe. But I know that some individuals have retained those extra digits into teenage and adult years. And I've actually had students who have acknowledged that that has been part of their genetic heritage, but in their cases have had the extra digits removed because they typically get in the way. There's very poor musculature and that sort of thing, and it just gets in the way. It's not an advantage to have it. Why 5 digits? We don't know. We really honestly don't know. But that seems to be the favored condition. But no one has really been able to point to a reason why it's the favored condition at this point. So if you want to take a look at this, you can read the section in the textbook. It takes a look at the type of fur that dogs can have and the different varieties of fur that dogs can have, and you can read up on that. What I do want to talk about, and I'm looking at the time here, and I know we're going way long and we've still got another section to look at yet, is that for individuals where there is the possibility of having a child with a genetic condition, where it is known that the genes are present in the family, or there is a suspicion of the genes being present in the family, there are ways of checking for it. You can take
You can access fetal cells through either amniocentesis, where you take fluid out of the amniotic sac using a very large needle, or you can reach in through the vagina into the uterus and collect some cells from what are called the chorionic villi that are also derived from fetal cells. That allows you to take a look at the baby's chromosomes from cells that are identical to the baby's cells, and you can screen for the presence of genetic conditions. And that then allows the couple to decide whether they want to carry the pregnancy to term or not, because there can be all sorts of complications as a consequence of different genetic conditions. It is also possible to capture fetal cells from the bloodstream, because fetal cells can escape through the placenta into the bloodstream. This is a very, very new technology, and it requires great skill to be able to isolate first the fetal cells and then the DNA from those fetal cells. When we get into chapter 11 and 12, particularly chapter 12, where we talk about DNA technology, we'll talk a little bit more about that, because it requires some very recent DNA technology to be able to do that. So this is the last break inserted into the presentation. And here are our concept check questions. So let me read these out, and then we'll stop for a quick break, and I'll meet you back on the other side of the break with the answers. So if two parents are carriers of albinism, what are the chances of their fourth child being albino if their first three are normal? What is the probability of a child inheriting a dominant trait from their parents if only one parent has the condition? See you on the other side of the break with the answers.